Hello everyone. Welcome to Enzyme MBBS, where complex medicine becomes simple and exam ready. Okay, so without wasting any time, let's get started. So, coming to the definition, phagocytosis is the process by which phagocytic cells, basically neutrophils and macrophages, ingest and destroy microbes, foreign particles, and dead cell debris as a part of innate immunity and acute inflammation. A pretty straightforward definition, it is self explanatory. Next, coming to the cells involved, as we know, it is neutrophils or macrophages, neutrophils in early responders and macrophages in later long lasting effects. Now, coming to the steps of phagocytosis. Now, this occurs in four sequential steps. And draw this diagram. This diagram is given in the Robbins, and it gives a very uh, extra edge for your answer. So, draw this diagram. And if you carefully observe, the diagram itself explains all the processes. So, first step, what happens? Recognition and attachment. The microbe comes and binds to the phagocyte receptor. Next, second process, it is engagement, or it is also known as engulfment. So what happens there? Basically, phagocytic membrane zips around zips around the microbe. So because of that, a phagosome is formed and microbe is ingested in it. And next, fusion of phagosome with lysosome. So there you can see inside a phagocyte there will be also the lysosomes with the enzymes. So that lysosome and this phagosome fuses to form phagolysosome. So this is the third step, and the fourth step, it is killing of microbes by lysosomal enzymes and reactive oxygen species and reactive nitrogen species. That is nothing but the last step, that is degradation of microbes by lysosomal enzymes in phagolysosome. Now we are gonna see all these steps one by one. So coming to the first step, that is recognition and attachment. What is the mechanism? It's nothing but microbes bind to the specific receptors on phagocytes. Now receptors, you can classify it into two types. One is opsonin receptor and one more is pattern recognition receptor. In opsonin receptor, basically opsonin coats the microbes which enhance the phagocytosis. If you have seen previous videos, especially the cellular mediators, not cellular mediators, the cellular events, there we discussed a scenario where a child is saying that I'm not gonna eat this. Okay, he's saying that I am not gonna eat this chapati. Now what his mother do? His mother makes rajma and gives him. Now that rajma's aromatic smell is like a eat me signal for him, and he goes and eats it. Right, similarly, opsonin gets bind to the microbes, and that is basically a eat me signal for the phagocyte, and it's gonna exhibit phagocytosis. So that is the role of opsonin. So those receptors are known as opsonin receptors. Now here I have given a table. It is nothing but the which opsonin is related to which receptor. It's nothing but a memorizing part, so just mention that also. So opsonin receptors are those receptors which perform most efficient phagocytosis. Coming to the second type of receptor, that is pattern recognition receptor. Now these recognize microbial component directly. If you carefully observe, in the opsonin receptors, the phagocyte was recognizing microbe with the help of opsonin, but here in the pattern recognition receptor, it's going to recognize microbe directly. So example for those kind of receptors are toll-like receptors, mannose receptors, and scavenger receptors. So basically they recognize something called as PAMs, so which abbreviates like pathogen associated molecular patterns. So what is the outcome? It is firm attachment of microbe to the phagocyte. You can refer the diagram also. Now second process, it is engulfment. So basically what happens, as we saw in the diagram, the pseudopods extend around the microbe and cell membrane will enclose it, and it basically forms a vesicle called as phagosome. And for this formation, actin polymerization is very much essential. I hope all know about actin, muscle physiology of first year. Okay, now coming to the third process, it is fusion with the lysosome. The phagosome basically fuses with the lysosomal granules, and that basically forms the phagolysosome. So lysosome provide enzymes and toxic substances. One point I would like to mention here is that in order to happen this fusion, lysosome basically requires a transporter. Okay, so that transporter is produced by lyst gene. This will be useful in future, so I have mentioned it now itself. Okay, Now coming to the fourth process, that is killing and degradation of microbes. It happens in a three ways. So one is a respiratory burst, where reactive oxygen species are involved, and one more is by reactive nitrogen species, and one more is by using lysosomal enzymes. Now here respiratory burst and reactive nitrogen species, they are basically oxygen dependent, whereas lysosomal enzymes, they are oxygen independent. So looking into the respiratory burst, so now oxygen is present, so oxygen is converted to O2 minus or superoxide by NADPH oxidase, and that particular superoxide is converted to H2O2 with the help of superoxide dismutase enzyme. Now H2O2, in the presence of myeloperoxidase enzyme and chlorine, it forms hypochlorous acid, whereas the same H2O2 in the presence of major basic protein and iodine, it forms hypoiodic acid. Hypochlorous acid formation is related to neutrophil, whereas hypoiodic acid formation is related to eosinophil. So this hypochlorous acid is basically bactericidal in nature, whereas hypoiodic acid is a parasite destructor. Okay, so and one more thing is that, see, chlorine, iodine, they are basically halogen, so that process is also can be referred to as halogenation. Fine. So the next thing is reactive nitrogen species. So INOS produces nitric oxide. So what is INOS? You can see it in the cellular mediators video. We have discussed that. So now nitric oxide formation takes place. So nitric oxide plus O2 minus, it forms peroxynitrate, which is basically toxic to microbes. Then the third method, it is using lysosomal enzymes, various enzymes like lysozyme, proteases, defensins, lactoferrin, acidic pH. All these facts play a role in phagocytosis or destruction of microbes. So what are the final outcomes of phagocytosis? It is killing of microbes, degradation of debris, clearance of necrotic tissue, and resolution of inflammation. Last but not the least, mention all the clinical significances here. So first thing is bystander tissue damage. It is, so during engulfment process, the phagocytes may release granule contents into extracellular space. So those substances can basically damage innocent surrounding normal tissue, hence it is known as bystander tissue damage. The second thing is Chediak-Higoshi syndrome. It is a kind of examiner's favorite. So yeah, it is a autosomal recessive disorder, and here basically mutation happens in LYST gene. So I mentioned during explaining fusion with lysosome, in order to lysosome fuse with the phagosome, lysosome requires a transporter. Now that transporter is coded by LYST gene. So now there is a mutation in it. So defective fusion of phagosomes with lysosome happens. Because of that, ineffective microbial killing happens. So what are the features you can see in the patient? You can see increased susceptibility to infections, and you can see leukocyte abnormalities, and here you can see albinism, peripheral neuropathy, and thrombasthenia. Now you may wonder, where did this albinism came, peripheral neuropathy came? The same transporter which helps the lysosome to transport, it is also involved in the transport of this one, melanin, as well as the for peripheral neuropathy, it is Schwann cells. So basically, when lysosome is getting affected, the melanin and your Schwann cells also getting affected. So because of melanin, albinism comes, and because of Schwann cells, peripheral neuropathy comes. And it is also something related to platelets, so thrombasthenia happens. The third thing is Bruton disease, or it is also known as X-linked A-gamma globulinemia. So in order to understand the pathology here, you have to understand the normal physiology. So what happens in normal mechanism? After antigen exposure, our B cells gets converted to plasma cells, and the plasma cell produce IgG or immunoglobulin G. So we discussed before in this video, immunoglobulin G acts as an opson in enhancing the phagocytosis. But now what happens in Bruton disease? In Bruton disease, the B cell maturation has a defect. So because of that, if B cell maturation is defect, then plasma cells won't be formed, and if plasma cells production hampers, then IgG production also hampers. So opsonin deficiency will be there. So consequences: defective opsonization, impaired phagocytosis, and recurrent bacterial infections. Fourth thing is chronic granulomatous disease. It basically happens due to deficiency of NADPH oxidase, which we saw in the respiratory burst. So that's all about the phagocytosis. I hope this was helpful for you.